discussion about CV may be tested on the exam like any other slide. Every lecture will be tested. ETC, signal transduction, and enzyme will be tested more. I will do an exam review next week, which will be more consolidated review. First of all, for carbohydrates, you don't need to know structure, but you should know what you are consuming in diet when you go to bake sales. For monosaccharide, you consume glucose, galactose, and fructose. You should know which one of them is the aldose sugar. Which is aldose sugar out of glucose, galactose, and fructose? Which one is an aldose sugar? Glucose, galactose, or fructose? Which one is an aldose sugar and not a ketose sugar? Glucose and galactose. Why I'm telling you to note that? Because foundation 2, there's a very important enzyme coming up, aldose reductase. So you should know what are your aldose sugar, glucose and galactose, but they are monosaccharides. Monosaccharide is the simplest and the only absorbable form for carbs. So you should know glucose, galactose, fructose are monosaccharide, not their structure, not their bonds, but they are monosaccharide. For disaccharides, you should know, let me get my laser pointer so we can have a better view. Disaccharide, you should know lactose and sucrose. Which one comes from milk and milk products? Lactose or sucrose? Which one comes? Lactose. Why it is important? Because in lactose intolerance, the milk and milk product containing lactose will lead to sign and symptom, not sucrose. Also, you should know what they are made up of. So what are your monosaccharides, disaccharides, sources? Monosaccharides are the best reducing sugar. They show up in urine. Most common sugar in urine is glucose. Other sugars are for foundation 2. But you should understand that monosaccharides are the best reducing sugar. That's why they give dipstick test positive in urine. Please know the sources. Poly, di, mono, keep them in under different class examples. Polystarch comes from plant. When you go to bake sales, you got to first understand what are you consuming. Secondly, you got to understand what type of source of a carb will cause signs and symptoms in which disorder. So you should first know this. Starch, plant. Glycogen, animal. Lactose, milk. Give me sucrose. How do you consume sucrose? Quickly. Sucrose. Sugar, table sugar. That's not the sugar grown on table, but the sugar that you consume. Monosaccharide. Glucose is not found free in nature. What's found free in nature? Fructose. How do you consume fructose? Fruit juices, and honey. Now, once you go to bake sale, you consume them. Know that amylase acts only on polysaccharides, on alpha linkage, not beta. Also alpha 1,4, not 1,6. Salivary and pancreatic amylase are not responsible for complete digestion. Where is complete digestion of carbs? In the small intestine, that releases monosaccharide, because this is the only absorbable form. Who will tell me why? Why monosaccharide is the only absorbable form? Unlike other macronutrients. Is there any reason that is the absorbable form? Because it's least osmotically active and inert form. Anything other than monosaccharide, for example, lactose intolerance, if lactose comes undigested in the large intestine, it's osmotically active disaccharide and causes havoc. So you should know that this is the only absorbable form. Secondly, amylase also helps you know clinical enzyme. Do you remember when amylase are raised? So what is the... Sorry? Please keep yourself mute. So when amylase is raised, what is the organ damage? So you should go back and always connect. So when amylase is raised, pancreatitis. So do your clinical enzymology there, clinical indicators of organ damage. Then you know that you went to bake sales, you know your sources, you know amylase does the digestion, but final digestion in the intestine. Now, who is ready in the lumen to be absorbed? Glucose, galactose, and fructose. Why? Because they are the only absorbable form. Are these three, glucose, galactose, and fructose, monosaccharide? Are these three monosaccharide? Yes, so they are the only absorbable form. Now you need to know the transporters. So there's an SG. So you need to know this flow. The sugars flow, monosaccharide flow from lumen to cell, cell to circulation, and from circulation, they are picked up by peripheral tissue. So let's see which transporters, we will do it in the interactive session, which transporters tell me pushes the sugars from lumen to cell. Which transporter is pushing the sugars from lumen to cell, glucose and galactose? SGLT1. Now, once you know SGLT1, do you know there's an SGLT2 there too? So what the question I asked is, tell me which transporter is pushing glucose and galactose from lumen to cell. I'm not asking from cell to circulation or peripheral tissue. I'm asking you lumen to cell. So you have to be specific. It's SGLT1. Now, who will tell me SGLT1 is a facilitated diffusion or secondary active transport? Mechanistically, this exam is important. So SGLT1 is a facilitated diffusion, secondary active transport by concentration of extracellular concentration of sodium, it's driven. But all the GLUT are facilitated diffusion, right? And since you have SGLT1, do you remember there's an SGLT2 too? Where is it? SGLT2. SGLT2. Kidney, renal, are there drugs that are SGLT2 inhibitors? So then bring the clinical application also. SGLT1, SGLT2. GLUT5 is always for fructose. Who is pushing all these sugars in the circulation? Which transporter is pushing all the sugar into the circulation? GLUT2, so they are specific. And then in the peripheral tissue, do you remember which is liver and pancreas? Who's pushing sugar in liver and pancreas? GLUT2 or 1 and 3? Liver and pancreas. 2, brain and RBC. Brain and RBC, 1 and 3. And did we do this by in insulin? The only transporter which is insulin mediated is which one? GLUT4. Where are they located? Where are they located? Skeletal muscle and adipose tissue. So see, you went to bake sales. You should know your sources of sugars. You digested them. Amylase acts on polysaccharide. Now, amylase acts only on alpha linkage. So can amylase digest cellulose? Because cellulose is also a polysaccharide. Fructose is always with 5. Okay, great. If you have something to remember that. Anyways, so I was asking you, amylase acts on polysaccharide, but only on alpha linkage. So can amylase digest cellulose? No, because cellulose has beta linkage. Absolutely wonderful. So you know there is a push of glucose after carbohydrate after digestion, which is the most absorbable form, monosaccharide. There is push of sugars from lumen to cell, cell to circulation, and then they are picked up by peripheral tissue. Are you clear with that? Yes, go back to your enzyme kinetics. Which one has a proportional flow, GLUT2 or 1 and 3? Who has proportional flow first order kinetics? 2 or 1 or 3? 2. Again, that's what happens. You go to bake sales, you collect carbs, glucose. Also, please know, though the absorbable forms are glucose, galactose, and fructose, but still, which is the most predominant sugar monosaccharide in your circulation after absorption? Which is glucose. That's why we talk more about glucose. Again, bake sales, glucose is high, GLUT2 proportional flow. Who is inside them? Who is inside them in liver and pancreas? Glucokinase. In pancreas, it will secrete what? After meal, what will be secreted? Insulin. Please don't do the mechanism here. I'm just trying to connect. Mechanism foundation 2. But I'm trying to connect. You go to bake sales. Monosaccharide, you digest carbs. Monosaccharide absorbable form. Predominant sugar, glucose. Glucose high after meal, proportional flow, GLUT2. Where does it go? Liver and pancreas. GLUT2 liver and pancreas. Who's sitting inside them? Glucokinase. In pancreas, glucokinase will release insulin. Now, insulin will bring down glucose to normal. Please don't do diabetic curve. This is all coming in foundation 2, but I like to connect, right? Then, once insulin is released, insulin is anabolic only to lipid and protein. What does insulin do? Turn on glucose utilization. Where is it? Foundation 2. What is foundation 1? Have we done all these signal transduction mechanisms? Foundation 1 is this box. Foundation 2 is the action of insulin. I did say insulin is anabolic to only lipid and protein on the right side, right? Did we learn that? So does somebody has a question. So does insulin have a growth factor like activity? Does insulin dark proteins GRB2 and has a growth factor like activity? So is this growth factor pathway RAS, RAP, MAP kinase pathway? Is RAS, MAP kinase pathway a growth factor pathway? Please know that, right? So this is all insulin action we have done. Have we done this PI3 kinase GLUT4 expression also? Have we done PI3 kinase GLUT4 expression? Insulin PI3 kinase. Have we done that insulin always and always lower cyclic AMP? Does insulin lower cyclic AMP inside the cell? And does insulin always dephosphorylates the key enzyme? So, and does insulin always dephosphorylates the key enzyme? Yes. So we have done all that. Now let's do questions. So mechanistically, you should know insulin mechanism of action. So what is this asking? Moving what? Glucose and galactose from where? Lumen to cell. So what would you pick? Glucose and galactose from lumen to cell. What should be your answer? Somebody's saying from lumen to cell, please. D, SGLT1, absolutely wonderful. From cell to circulation will be GLUT2, but lumen to cell is SGLT1. Is it a secondary active transport? Absolutely. And is there SGLT2? Absolutely. Where is SGLT2? Renal. And there we have done SGLT2 inhibitors. Dr. Binstock has introduced that. Now look at this. Pancreatic function is not working, right? So tell me, if I didn't tell you to know what are your mono, di, and polysaccharide. Now, if pancreatic enzyme is not working, will you even choose A, B, or E? Do they need to be digested? A, B, and E? Do they need to be digested? No. Why? Why A, B, and E don't need to be digested? Because they are monosaccharide. So pancreatic efficiency has nothing to do with it. What about cellulose? Why wouldn't you not choose cellulose? Is cellulose digested by pancreatic amylase? No. So that is why if pancreatic enzyme is not working, what will be undigested? Starch or any polysaccharide, but not the others. Now, what is lactose intolerance? Please see few clinical and both specific importance of lactose intolerance. First of all, deficiency of which enzyme? Lactose is deficiency of which enzyme? Lactase. Lactase is a disaccharidase. It has a specific location. It's located at the brush border of epithelium. So please know that clinically lactose intolerance is not just primary, it could be secondary because of loss of lactase enzyme, which can happen where? How can you lose lactase enzyme? Can you lose lactase enzyme and have secondary intestinal injury, secondary lactase intolerance, secondary lactase deficiency, intestinal injury, infestation, and any kind of GI infestation? Somebody says, okay, I was lactose intolerant until I had a GI infestation. Age, very good. With age too. Sometimes with age, you see, I could tolerate lactose, not now. But please know that also, please understand, students get confused. Amylase digest only alpha 1, 4. But for disaccharides, there's nothing called alpha beta. So don't be confused. In disaccharides, there's no distinction or specification of alpha versus beta. They can do both. But now if lactase is deficient and lactose come undigested in the large intestine, this is a havoc. Because this disaccharide is now what? Why is this dangerous? Lactose not be digested and come undigested in the large intestine. What is going to happen? What is it? What is it specifically now? It's osmotically active. It will draw in water, watery diarrhea. Yep, because it's osmotically active. Any bacterial fermentation will give you the fermentation product that will cause pain, abdominal pain, bloating with floating. Remember, hydrogen is also a part of fermentation product. That's why, do you know the non-invasive test for lactose intolerance is hydrogen breath test. So this is all about lactose intolerance. Also, please know there you can also give oral lactose and watch the fun, but that's not good, right? So that's why it's a non-invasive hydrogen breath test is done for lactose intolerance. So you understand what is lactose intolerance? Lactose should be digested. So what will be lactose digested into? What should be the lactose digested into? Which monosaccharides? Glucose and galactose if lactase was present. But since lactase is deficient, lactose comes undigested in the large intestine, osmotically active, draws in water, fermentation. Fermentation products cause pain, cramps, bloating, and hydrogen breath test is non-invasive test. And looking at the age of the child, would you pick, and also please, there should be a history of what? That's why I asked you to know the source. In lactose intolerance, intake of not fruit juices and table sugar, but milk products will lead to what? Digestive symptoms like pain, bloating, and cramps. And so the history of source is important. So because of age, you will pick primary or secondary because of age? Primary. So you know what is lactose intolerance. Give me a minute breather, will start exam review, right? Okay, everyone. Now, for the exam review, please know that I'm not teaching. I'll tell you, please note down this. If you're not note down, please, you have enough of time. You have weekend. Also see that I'm opening because since students are learning a lot and doing their stuff. On Sunday, I will open 2 hours for question and answer. You can log in, but it will be enabled. Few people can only log in because I want to use some one-to-one interaction. Keep room for that. I'll send out the timings for Sunday. Is that okay? Right? Great, because I think it's a good time to answer your questions. Now, first of all, let's do overview. Metabolic map will be given in your exam. Not very useful. Foundation 1, Foundation 2. Please note down. I'll be a little fast in this. I will do individual office hours. I'm also available 12 to 2 today. Please stick to the learning objectives. You should know anabolism versus catabolism. So which one needs ATP, acetyl-CoA, NADPH, insulin mainly, mostly? Which one? Catabolism or anabolism? Anabolism. So you got to clearly distinguish that. Now, catabolism. Is it by this big enzyme oxidoreductase and subclass dehydrogenase? Keep an eye on that, please. Why? Because they all need vitamins as coenzyme. So you got to keep an eye on which vitamin is which coenzyme. Foundation 2, we will connect where they are used. Please see, I will be quick. You could note down. And today also 12 to 2, I'm open. You can walk in. I'm in the office or Zoom with me. Then comes the multi-step enzyme catalyzed pathway. Please know that the which steps are regulated, reversible or irreversible. Which are the regulated step? Reversible or irreversible? Irreversible step. They could be rate limiting and committed, but they are all subjected to regulation. Now, which one of them alters enzyme activity, long term or short term? Which one is short term? So you got to be clear with the mechanistic of mechanism and basic in this exam, but also connect clinically at the end. Don't forget that. So you're very well aware of long term versus short term. It's the short term that affects enzyme activity, not long term. Also, please know some of the examples. Substrate concentration. We just did that. After bake sale, is glucose high? Does it flow through GLUT2 and inside is glucokinase? Yes or no? Did I tell you that? You go to bake sales, glucose is high, proportional flow, GLUT2, glucokinase is sitting there. So imagine, is glucokinase affected by glucose concentration, which is substrate concentration, which is the first one? Yes, glucokinase cannot be active during fasting. Please know that. It's active after meals. Now, somebody asked me what is proenzyme into enzyme conversion. Please know that some pancreatic proteases cannot be active inside the cell. Why? Why can some pancreatic proteases, example, be active inside the cell? What will happen if they are active inside the cell? It will cause autocell lysis, autodigestion, and pancreatitis. That's why they exist as proenzyme in the cell and they become active only in the lumen as enzymes. Proenzyme into enzyme is that kind of regulation. Somebody asked me compartmentalization. Please know that it will come in Foundation 2 very easily. Compartmentalization means some processes are cytosolic, some are mitochondrial. For example, oxidative phosphorylation is cytosolic or mitochondrial. Oxidative phosphorylation is cytosolic or a mitochondrial process? A mitochondrial process. That's also part of compartmentalization. More will come up. So we distinguish long-term from short-term regulation. This is what I was telling you, that glucokinase cannot be active in fasting. It's only active after meals. Sometimes students look at ATP only as allosteric modifier. ATP has various roles in the body. One of them is giving phosphate for phosphorylation, back dephosphorylation. Did we learn insulin always and always? Insulin always and always? Phosphorylates or dephosphorylates the key enzyme? Dephosphorylates the key enzyme. And what protein kinase did we connect here with signal transduction? What protein kinase did we? Protein kinase A, not B. Where was B? Where was protein kinase B? Protein kinase B is with which ligand? Insulin. So you got to distinguish your protein kinase. So keep connecting overview with signal transduction and understand that. Enzyme kinetics. As I told you, don't jump to the inhibitions, clinical examples. First, enzyme protein, non-protein parts. So non-protein part as coenzymes, what are the examples? Who are coenzymes in your body? Are there coenzymes in your body? Are there anything, nutrients that act as coenzyme? Which nutrients act as coenzyme? Vitamins. Keep a close eye on your vitamins. Yes, enzymes act by lowering activation energy. But what about this overall Gibbs free energy? Is it altered by the enzyme? No. So some things are altered by the enzyme, some are not. Keep a close eye on that, please. Also, we only did a big class oxidoreductase. What's the subclass you should know there? We did a big class oxidoreductase. What's the subclass? Dehydrogenase. Very good. Give me the coenzymes dehydrogenase uses. What coenzymes do they use? Quickly, NAD and just NAD? FAD2. Dehydrogenase use both NAD, FAD, and even NADP. Even NADP. Sorry. I don't want to go the NADP because that will be Foundation 2. Yes, whatever we have learned for oxidation, let's concentrate on oxidation. Dehydrogenase use more NAD than FAD. Are you clear with that? That dehydrogenase use more NAD than FAD. Are more substrate linked to NAD versus FAD? Are more substrate linked to NAD? Yes, just know that. Clinical enzyme is important. You got to understand specificity. For example, did I tell you that amylase could be pancreatic origin? What about cardiac? In cardiac origin, would you talk about ALT or would you talk about CKMB? Cardiac damage, myocardial damage, CKMB. So you just got to be specific here, please. You just got to be specific here. Great. So what happened, right? Okay, sorry. You got to be specific here. What organ damage leads to what enzyme or isoenzyme rise? Now, some students are confused with that. So now understand that enzymes are within the cell. Whenever there is breach in cell lining, these enzymes leak into the circulation and show above normal range. So that's why whenever these rise above normal range in the circulation, they indicate specific tissue damage. For example, LDH1 or 5, which one is for specific for cardiac damage? 1 or 5? 1 or 5? 1. Now you don't have, now know that you should know the normal first, then go to inhibition. I told you that there are allosteric and non-allosteric enzyme. Who follows Menton kinetics? Allosteric or non-allosteric? Who follows Menton kinetics? Non-allostric enzyme. Inhibition is explained by non-allostric enzyme. First of all, you should not that you should memorize this equation, but you should know what is VO, initial velocity, no product inhibition. You should know what's max saturation. What is saturated here? Enzyme or substrate? What is saturated here? Enzyme. So Vmax can only be altered by altering the number of enzyme. If Vmax moves down, are you decreasing the number of enzyme? Then Vmax goes down, or you increase the number of enzyme that Vmax goes down. When you decrease the number of enzyme, please know that this is saturation. Also know that initial part of the reaction, there is proportional change, and this is extrapolated. Glucose transporters also follow enzyme kinetics where they show proportional flow. Who shows proportional flow? GLUT1, 3 or 2? 2. GLUT2 is where? Liver, pancreas, or brain and RBC? GLUT2. GLUT2, liver, pancreas. What's sitting inside them? What's sitting inside them? Liver and pancreas enzyme, glucokinase. Don't mix and mess that up, right? On this part of the reaction as larger than Km, this is zero order kinetic. The velocity is independent of substrate concentration. Who extrapolates the same kinetic zero order? GLUT1 and 3. So 1 and 3 show constant flow or proportional flow. 1 and 3. Constant flow. Where are they? Brain in RBC. What's sitting inside them? Most of the tissue, hexokinase. So that glucokinase, hexokinase comparison is important. Please know that. Now, right shift or left shift. Why right shift of the hyperbolic curve shows inhibition? Why right shift shows inhibition? Why does? Because you need more substrate concentration if you shift the curve right to reach the same Vmax by 2. You need more substrate concentration to reach the same Vmax by 2. Now, the thing here is you should know that what Km reflects. Is Km on x-axis or y-axis? Is Km on, it's on x-axis. So Km reflects affinity or efficiency of the enzyme. Km affinity. Low Km means high affinity. Vmax reflects affinity or efficiency. Vmax reflect efficiency or efficiency. Can Vmax be altered by the number of substrate concentration? Is Vmax dependent on substrate concentration? No. Then you go to this line Weaver-Burke plot. You know the x and the y intercept. So if this straight line is moving right, is Km increasing or decreasing? If the straight line is moving right, Km is increasing. Where does Km increases? Competitive or non-competitive inhibition? Competitive inhibition. If this straight line is moving up, is Vmax increasing or decreasing? It's decreasing. Where is Vmax decrease? Competitive or non-competitive inhibition? Non-competitive inhibition. Please always, inhibitor line is always above the control line. So the terms, what do they stand for? What does Km reflects? What does Vmax reflect? That is more important to you before you come to the extrapolation or enzyme kinetics and inhibition. I have explained you all first order, zero order. This is mixed order. And Vmax is independent of substrate concentration. Once you reach saturation, Vmax can be altered by increasing substrate concentration. You're very well aware of Km. If you shift the curve right, hyperbolic curve right, do you need a larger substrate concentration to reach the same Vmax by 2? If you shift the curve right, do you need a larger substrate concentration to reach the same Vmax by 2? So moving the hyperbolic curve right is activation or inhibition? Moving the hyperbolic curve right is activation or inhibition? Inhibition. Is it competitive or non-competitive inhibition? Not competitive inhibition. Very good. I like that. You know glucokinase and hexokinase. Now, students are very confused with that. Please see the reactions. All these will come in foundation too. You got to go mechanist. So let's see this, that how will you memorize glucokinase versus hexokinase? Now tell me, glucokinase is in liver and pancreas, right? Liver is the chief site of metabolism and pancreas help in secreting insulin. So what enzyme you want in liver and pancreas? An enzyme of good efficiency or good affinity? What is your priority here? Liver and pancreas. Good efficiency. That is why glucokinase has what? Who reflects efficiency, Vmax or Km? Who Efficiency. Efficiency is Vmax, not Km. That's why glucokinase is with a high Vmax is in liver and pancreas, irrespective of Km. Glucokinase has a high Vmax as well as high Km, but your need is that you need an enzyme of good efficiency, which is reflected by high Vmax in liver and pancreas. So that is why glucokinase has a high Vmax and high Km. But look at brain and RBC. In brain and RBC, you cannot divide them any time of glucose utilization. So do you need an enzyme of high affinity or high efficiency here? High affinity. High affinity means high Km or low Km. High affinity means low Km. That is why hexokinase is an enzyme of low Km. That's what you need in brain and RBC, and its Vmax is also low. So if you do compartmentalization, which somebody was asking, and purpose, you will know difference between hexokinase and glucokinase. I'm finishing irreversible here only. Irreversible and suicidal graph looks like competitive or non-competitive. Irreversible and suicidal graph looks like non-competitive. But there is a difference. Whenever they say irreversible, inhibitor binds to the enzyme. Now you got to be very careful. I hope you are following my pace because I got to finish everything, right? Is that okay? Are you noting down? Everything is good? Great. Please know that also know that inhibitor does not bind to the substrate. You should get used to this language that inhibitors binds to the enzyme and change the kinetic of the enzyme. We will see what's the change of kinetic of the enzyme in the presence of competitive versus non-competitive inhibitor. But in irreversible inhibition, inhibitor binds covalently to the enzyme. That word is important for you to pick irreversible. How does irreversible become suicidal? Sometimes an inhibitor binding to the enzyme also kills the enzyme. That is why irreversible can be suicidal, right? Please, not just why this slide is there, not just knowing the graph, also understanding through graph, although I told you when it's hyperbolic curve, see what is changed. In double reciprocal plot, quickly see what is unchanged, but also do the kinetics. For example, when you say competitive inhibition, is there a similarity between structure, substrate and inhibitor? Is there a similarity between inhibitor and substrate? I'm just giving you one example. In competitive, yes. Is there decrease in affinity of enzyme for the substrate in the presence of inhibitor? Is there decrease in the affinity of the enzyme for the substrate in the presence of inhibitor? That increases Km. Can you overcome the inhibition by increases substrate concentration? That increases Km in competitive inhibition. And since you can overcome the inhibition, do the number of enzymes practically, the outcome is remain unchanged in competitive inhibition. Do the number of enzymes remain unchanged outcome? That's why Vmaxes remain unchanged. So a kind of background and a little why. But quickly, graph, you should be familiar. So what is changing here? Vmax or Km? What's changing here? Vmax or Km? So is it competitive or non-competitive? Km, and Km increases in competitive inhibition. In double reciprocal plot, you will see what is unchanged. Is x-intercept unchanged or y-intercept unchanged? X or Y here? Y. Y reflects Km or Vmax? Y-intercept reflects Km or Vmax? Vmax is unchanged in competitive or non-competitive inhibition. Vmax is unchanged in competitive or non-competitive inhibition, competitive inhibition. So yes, and always, please, inhibitor line is always above the control line. Don't make that mistake, all right? So do kinetics. Now, for example, if you know the reasons, do you see a ESI complex in competitive inhibition? Do you see a ESI complex, enzyme substrate inhibitor together? No. You see that in non-competitive inhibition, because in competitive inhibition, inhibitor binds to the same site as substrate binding site. Do the same for non-competitive inhibition. As I told you, in hyperbolic curves, see what is changed. In double reciprocal plots, what is unchanged to get to the answers quick. I've explained you all. This is the summary. You should know Vmax, first order, zero order, sorry, right shift, left shift, up and down, Also, please, why Vmax decrease is repression? What is this under? Vmax decrease, is it repression? What is it under? Which regulation? Long term or short term? Vmax decrease is repression because it's under long-term regulation. You decrease the quantity of enzyme. Absolutely wonderful. So please do competitive versus non-competitive inhibition. I explained you that suicidal inhibition usually when the inhibitor binds to the enzyme, kills the enzyme. Please, this is not for you to know, and competitive inhibition is not part of your course objective, but clearly by this graph and parallel lines, you can say what's happening to both Vmax and Km. Km is going left and this line is moving up. So what is happening here? Both to Km and Vmax, are they increasing or decreasing? Both are decreasing. Km is moving left, Vmax line is moving up. So both Km and Vmax are decreasing. So with the help of graph, you should not be confused. Where would be the line moving, whether hyperbolic or double reciprocal, when there are changes in Km and Vmax. Clearly, when it's velocity versus substrate concentration curve, the curve should be hyperbolic or sigmoid. What should be first the curve with to call it allostric? It should be hyperbolic or sigmoid? Sigmoid. You should know properties of allostric enzyme. Since there is a smooth coordinated change, that's why the curve is not a straight line but S-shaped. Allostric enzyme, do they follow Menton's kinetics? Do allostric enzyme follow Menton kinetic? No. Do they have a Km? Do they have a Km? No, but they are K and V type. If S-shaped curve is moving right and left, K type. If S-shaped curve is moving up and down, V type. Now tell me, S-shaped curve moving right or left, what is inhibition? S-shaped curve moving right or left? Right is inhibition, right? Does that make sense? So understand by graphs. Also, don't do calculations, structure, bonds, but understand what is Km. Km is substrate concentration at which reaction is Vmax by 2. Km is on the x-axis. Km is constant. Km reflects affinity of the enzyme. Low Km means? Low Km means? Low Km means high affinity. Who's the high affinity enzyme? Hexokinase or glucokinase? Who's the high affinity enzyme? Hexokinase or glucokinase? Hexokinase, absolutely wonderful. You're doing good. Now here also you're not doing calculation, sorry, but you are trying to understand when S is equal to Km, it's Vmax by 2. When S is larger than Km, is it first order kinetics or zero order kinetic? If S is larger than Km, is it first order kinetics, zero order kinetics? And what have you reached? What have you reached? Vmax. For a Vmax. Vmax is saturation. Vmax cannot be altered by substrate concentration. Primers are not tested on the exam, but I want you to know what is G, Gibbs free energy, difference in the energy of products to that of, products to that of reactants. Is it altered by the enzyme? Is Gibbs free energy altered by the enzyme? No. What is this energy? Energy released when you do oxidation. Please see, I will try to cover as much as I can for today. I'm still going at a fast pace, but I'm going to do another review on Monday or Tuesday. We can finish it, right? I hope you're following this pace. Everybody's good? Is it helping? It's taking a breather for me. Yes, less tense now. Good, very helpful. So I'm going to see how much we can finish by 10:50, right? I think we should be done. Again, so you should know what is overall Gibbs free energy. Gibbs free energy change is difference in our energy of product to that of reactants, not the other way around. It's not altered by the enzyme. It is released by catabolism, oxidation. Is oxidation exergonic or endergonic? Is oxidation exergonic or endergonic? Exergonic, and that energy is trapped in the form of ATP, for which you need ETC. Oxidation, new definition is loss of what? What is the new definition of oxidation in your body? Loss of 2H plus and 2 electrons, which is known as reducing equivalent. Who's the reflection of ATP, NADH or NADPH? NADH or NADPH? NADH. Please know that macronutrients are oxidized and substrate are reduced. Major oxidation pathway, TCA cycle, that will come in foundation 2. So most of your reduced coenzyme after oxidation are lying where? Which compartment are they lying? Most of your reduced coenzyme after oxidation, NADH and FADH2 are present where? In the mitochondrial matrix. What's present in the inner mitochondrial membrane? The ETC, which can quickly convert these reducing equivalents into ATP. So know that oxidation is coupled with phosphorylation. This is known as oxidative phosphorylation. Some people are confused. What is the difference between oxidative phosphorylation and substrate level phosphorylation? Which one, oxidative or substrate level phosphorylation? Which one you need ETC to produce ATP? Oxidative phosphorylation or substrate? That's the difference. Oxidative phosphorylation means you need ETC to produce ATP, number one. Secondly, please know your vitamins and coenzyme. If the ETC is not functioning, any inhibitor. If mitochondria is not functioning or absent, or if oxygen is low, can you do oxidative phosphorylation? Can you do oxidative phosphorylation if ETC, mitochondria, and oxygen is missing? No. TCA is the main cycle, which is giving you NADH and FADH2. They have to go where to produce ATP? Where do they have to go? Where do these NADH and FADH2 have to go? ETC, electron transport chain, all the components. But please know what are shuttles. Since most of the NADH and FADH2 are in the matrix, what will happen to the NADH outside the matrix in the cytosol? For them, you need shuttle. You don't need to know details of the shuttle coming in Foundation 2, but since I have mentioned in my lectures, I'm just telling you shuttles are needed for cytosolic NADH. All the NADH and FADH2 in the matrix, no worries because ETC is in the inner membrane. 5 fixed complexes. Are you seeing ATP synthase complex? What is it also known as? ATP synthase complex is also known as F0, F1 complex, please. FO, F0, F1 complex, right? Who helps in synthesizing ATP? Complex 5. Where is ATP synthesized? All except, where is ATP synthesized? By all complexes except 2. Why 2 is not a coupling site? Does that make sense? So know what are they, know what are they made up of, then know what is the flow. Please be very mechanistic here. So more substrate are linked to NAD, less substrates are linked to FAD. Each complex has different component. Complex 5 is shown here. Is complex 5 shown in this slide? No, complex 5 is also known as F0, F1 ATP synthase complex. Also, since this complex is linked to oxygen, what is this complex also known as? Complex. This last complex is known as complex what? This complex. Complex 4, also known as cytochrome oxidase. Tell me its inhibitors. So first you understand the components, flow, then inhibitors. Inhibitors of complex 4, carbon monoxide and cyanide. Absolutely wonderful. Also know function of each complexes. Know the flow is unidirectional. It's not back and forth. What's the function of complex 1? Tell me. Passing reducing equivalents from where to where? What's the function of complex 1? To pass reducing equivalents from NAD to CoQ. That is why, tell me if complex 3 is inhibited, what will happen to CoQ? Will it be in oxidized state or reduced state? Complex reduced state. So first you got to go this. Then you go to inhibitor and effect on 3. Very important. Number 1, ATP synthesis. Number 2, ETC function. And number 3, oxygen consumption. You go there, right? Does that make sense? 2 is not a coupling site. Now, one confusion also students have that the 2H plus an electron either linked to NAD or FAD, they have to go right up to the oxygen. But remember that electrons participate in the component. but protons have their circuit. Now, somebody has asked me redox potential. Please don't go deep into redox potential. Redox potential only shows affinity of electron for the system. So tell me, do you want electrons to easily flow from the substrate NAD right up to oxygen? Do you want? Yes. So that is why all the electron transport chain components are arranged in the order of increasing redox potential for easy flow of electrons. So who should have the highest redox potential? Who should have oxygen? Absolutely wonderful. Hydrogen do not flow as electron. Electrons participate with the components of complexes. Hydrogen follow a circuit. So are all the reduced coenzyme in the matrix? Are all the reduced coenzyme in the matrix after oxidation, because TCA cycle is in the matrix. So the flow of proton is from matrix through these complexes. Is 2 allowing protons to flow? Is 2, complex 2 allowing the proton to flow here? No. That's why complex 2 does not synthesize ATP, because only when proton flow from matrix to intermembrane space back to FO-F1 complex, then you synthesize ATP. All I'm bringing to your attention is first no normal, then no abnormal. Then bring in inhibitors. What will happen if you inhibit complex 1, complex 2, 3, or 4? Understand that whether electrons move by participating within the components or proton follow these circuits, both have still to go finally where? To oxygen to form what? 2H plus and electron have to go to oxygen to form water. Secondly, go mechanistic. So do the inhibitors of electron transport chain uncouplers or oligomycin? Who acts by preventing the proton flow? Who acts oligomycin? So they have a very particular through the FO-F1 complex. So what do uncouplers do? Also, inhibitors and oligomycin, do they have the same outcome? Inhibitors and oligomycin, ETC inhibitors and oligomycin, do they have the same outcome on ATP, ETC, and oxygen consumption? Do oligomycin and ETC inhibitors have same outcome on ETC function, ATP synthesis, and oxygen consumption? Absolutely yes. Who is different? Uncoupler. What does uncoupler do? Shuttles. Increase permeability of inner mitochondrial membrane to H plus ions. Normally, inner membrane is not permeable to H plus ions. Is that good? No. Is that good? Why? Why increasing permeability by uncoupler is not good? What will it abolish? This proton circuit. This proton circuit. So uncouplers, 2,4-dinitrophenol, 2,4-dinitrocosol, thermogenin, aspirin in high doses, all do what? ATP synthesis, uncouplers. What is happening to ATP synthesis? Uncouplers. Increase or decrease? Down. ETC keeps going on and on and on and on like a spoiled child. And oxygen consumption keeps on increasing. And since you're not trapping the heat in the form of ATP, are uncouplers hyperthermic? Are uncouplers hyperthermic? Yes. So please know uncoupler dissociate oxidation from phosphorylation. You should know very well ATP is the chief controller of ETC. Do you lose ATP control in the presence of uncouplers? Is ATP control on ETC lost in the presence of uncoupler? Is the proton circuit abolished in the presence of uncoupler? So please go mechanist. Please understand the mechanism and these 3 outcomes. Secondly, give me an inhibitor of complex 2. Is it antimycin or malonate? Malonate. So you got to first be specific there, right? Carbon monoxide and cyanide, which is reversible. Both bind to complex 4, carbon monoxide. Cyanide is irreversible, which needs amyl nitrite as an antidote, which needs cyanide poisoning, which has closed apartment, burning furnaces in closed apartment history, carbon monoxide, automobile pollution. Understand uncouplers. Thermogenin is also an uncoupler. Uncouplers have a different outcome than ETC inhibitors and oligomycin. They are very specific, please. Please do these questions. I will bring them in the whiteboard. Please see, this is an extra help. I've already told you what to do. I may bring this back, but first do it. I'm going to post these slides, do these questions. I'm going to bring this in the next exam review. Now let's see how much we have 10 minutes. We can easily complete signal transduction. Yes, are you ready? Signal transduction, how would you go about that? Signaling molecule, receptor, signal transduction mechanism. Don't miss any link. Signaling molecule, hydrophilic, lipophilic. Which one uses intracellular receptors? Which one uses intracellular receptors? Lipophilic, keep them separate. They work at the level of genes, steroid, thyroid. Then hydrophilic messenger, they use plasma membrane receptors. Please divide them into three types: heptahelical, tyrosine kinase, serine threonine kinase. I will tell you now which one to do in detail. Heptahelical in detail. What are heptahelical associated with? Trimeric G protein or monomeric G protein? Heptahelical, trimeric G protein, GS, GI, and GQ. Please identify second messengers of GS, GI, and GQ. Now go to tyrosine kinase receptor. Growth factor use intrinsic tyrosine kinase receptor. Insulin uses a separate tyrosine kinase model. Please don't do in detail cytokines, which uses associated tyrosine kinase receptors. Serine threonine is TGF beta. Please don't do that also in detail. Cytokines and TGF beta, not in detail, but serine threonine kinase is TGF beta. So first segregate lipophilic, intracellular messenger, then do hydrophilic. Please divide their receptors into heptahelical. Do heptahelical first in detail, then tyrosine kinase in detail. In tyrosine kinase, do growth factor and insulin in detail. Identify them step by step. For example, when you do epinephrine beta, GS stimulatory, what comes first, adenylyl cyclase or pKa? When you do epinephrine beta GS, what comes first, adenylyl cyclase or pKa? You got to be mindful with that step. Yes, it's adenylyl cyclase. Then adenylyl cyclase forms cyclic AMP or phosphodiesterase. Adenylyl cyclase forms cyclic AMP or phosphodiesterase. Who forms cyclic AMP? Adenylyl cyclase protein, membrane-bound protein, or phosphodiesterase? Adenylyl cyclase, you got to be specific. Glucagon is always working with epinephrine, similar to epinephrine beta GS, but using glucagon receptors. Where are glucagon receptors absent in the, where are glucagon receptor absent? Very important for foundation 2 muscles. So do GS, GI, and GQ. They have different second messengers. Insulin acts by increasing or decreasing cyclic AMP. Insulin acts by increasing or decreasing cyclic AMP. Insulin act by decreasing cyclic AMP. Please know that. pKa is an allosterically enzyme. So tell me, pKa is an allosterically enzyme. Who activates pKa? Epinephrine beta or GS or GI? Who activates protein kinase A? Epinephrine GS or epinephrine GI? Epinephrine GS. It's an allosteric enzyme. Cyclic AMP is its positive allosteric effector. Also, please, now don't forget the clinical. Now, somebody has a question. I think we'll be able to finish all the slide. Somebody has question when the RAS. Please know that RAS. Okay, I'll bring RAS later on. It just came to my mind. Now do clinical application of GS and GI. Tell me, pertussis acts on GI or GS? GI. Pertussis acting on GI, modifying GI, make it persistently active or inactive? Inactive. Do the other for cholera. Okay, does that make sense? Now, GQ. When you do GQ, do you talk about cyclic AMP or IP3 and DAG? When you do GQ signaling, do you talk about IP3 and DAG? Please know protein kinase C comes at the end. So protein kinase C is here. Where is protein kinase B? This is C. Where is protein kinase B with which ligand? Insulin. So differentiate that. Please know that IP3 moves calcium from inside to outside, from ER to, sorry, inside, and DAG moves extracellular calcium inside. So in the end, tell me, if it's GQ signaling, what will be active? If GQ signaling is active, give me the last protein that will be active. Last two enzymes that will be active. If GQ is active, give me the last two enzymes that will be active. Now, don't say IP3 and DAG. That's what I want you to distinguish. IP3, DAG are not enzymes. They are second messenger. You got to be mindful of that. Yes, somebody answered very specifically PKC and calcium calmodulin kinase. They are the enzymes. Differentiate enzymes and second messengers. PKC and calcium calmodulin kinase should be active at the end, right at the end of GQ signaling. So please fill it up, right? You have, please know that CV may or may not be tested, but it's part of interactive session, right? This is basic, and we have introduced drug. So know where these drug function. For example, is albuterol beta antagonist or beta agonist? Antagonist or agonist? Albuterol, agonist. That's why it will stimulate which pathway? So it will stimulate what? GS or GI? GS. So you have to have the basic knowledge to get to the drugs. Does that make sense? Also, please, physiology will teach you in detail. We are not testing you. I'm so sorry, I'm exhausted. Physiology will teach you in detail, but not us, that though albuterol is beta agonist, it still has different effects on different cells, not us. For tyrosine kinase, don't forget intrinsic tyrosine kinase, growth factor in detail. JAK-STAT serine kinase, not in details. Now here somebody asked me, let's finish this. I think I'm almost at the end. Give me 2 minutes, okay? Please know, so growth factor uses beta adrenergic heptahelical receptors or tyrosine kinase. Growth factor uses heptahelical receptors or tyrosine kinase. Which receptor? Tyrosine kinase. Also, it's associated with this RAS, RAF, MAP kinase pathway. Please bring it all so that you know it's a total RAS, RAF, MAP kinase pathway. Please know RAS is bound to GTP for normal growth. But if RAS is persistently bound to GTP because of mutation, what will happen? What will it lead to? Cancer, not otherwise. Otherwise, RAS is bound to GTP and does growth. Does that make sense? So when RAS is persistently bound to GTP, then there would be cancer. That's pathological. Insulin also follows its own tyrosine kinase model. Please be mindful. Insulin is diverse and docks different protein for different action. So tell me, GRB2 or PI3 kinase, which protein insulin will dock for growth factor-like activity? GRB2 or PI3 kinase, GRB2, and that's the RAS, RAF, MAP kinase pathway. For PI3 kinase, if insulin is docking PI3 kinase, what is it increasing? If insulin is docking PI3 kinase, what is the outcome? GLUT4 expression. Do that also with PI3 kinase. What protein kinase adds on for GLUT4 expression? Is it A, B, C, or G? B, very specific. Do GLUT4 then do GLUT4 with your carbohydrate digestion too. Every signaling molecule has a termination mechanism. If you saw epinephrine beta, the termination was epinephrine alpha 2. If you saw increase in cyclic AMP by which hormones? Increase in cyclic AMP inside the cell is by which hormones? Or which ligands? Which ligands increase cyclic AMP inside the cell? Epinephrine and glucagon. Did you see that there is one ligand that decreases cyclic AMP? Who decreases cyclic AMP? Insulin. So for every signal transduction mechanism, there is a termination mechanism, please. Don't do triggers now. They are very important for foundation 2. I always work ahead. Protein kinase G is nitric oxide. Sildenafil will come in foundation 2 with Dr. Binstock's questions or lectures. For every signal transduction mechanism, please know that there is a termination. I wish you good luck.
Thank
Great.
you.